Hello everyone, I am Alejandro Torres. I'm from Oklahoma State University and I'm very eager to participate in this conference and share my work with you. In the United States, 9.5% of high school age students develop chronic oxycodone use. The brain goes through neuroadaptation since infancy, and adolescence is a critical period, highly active in neuroadaptations that will continue until adulthood. The opioid addiction cycle starts by using, leading to dependence and addiction, and when the drug is stopped, emotional and physical effects of withdrawal account for the relapse and the continuous cycle. Also, adolescent gut microbiome is affected by opioid usage, and the most common side effect is constipation. In adults, microbiome gut-brain axis contribute to changes in cognitive function and mental health. The brain communicates with the gut microbiome in a bidirectional way, influencing the behavior, the mental health, the pain, and neurogenesis. So, is the gut microbiome critical for shaping adolescent brain and behavior? Brain-derived neurotrophic factor, or BDNF, is a neurotrophic that is important for neuroplasticity, and it is found in two forms, pro-BDNF and mature BDNF. For example, physical exercise, social interactions, and a healthy diet increase the mature BDNF levels, which stimulate neural survival, efficient pruning, and synaptic plasticity. In contrast, stress, maternal deprivation, alcohol, and heroin use increases the pro-BDNF levels, which induces apoptosis, neuroinflammation, and atrophy. BDNF from the gut microbiome modulates neurotransmitter release in the enteric nervous system and affects the motility in the colon. Our specific aim encompasses to understand the mechanisms of social stress affecting the microbiome dysbiosis, modulation of BDNF, and how it impacts the oxycodone and hedonic effects. Our experimental approach of ACEs, adverse childhood experiences, aims to mimic a neglected household dysfunction environment using a translational animal model with Sprague-Dawley rats. For example, our isolated environment will be socially neglected, there will be no motor or sensory stimulation, and our enriched environment will receive motor, sensory, and social stimulation. And also we'll compare that with a controlled and startup environment to investigate how ACEs and opioids affect the brain and gut microbiome in the adolescent. Our differential reading environments are going to be exposed for a period of 30 days in the same condition, and then on the day 52 they will begin receiving a dose of oxycodone treatment twice a day in incremental manner, and on the last day they will receive an naloxone injection that will precipitate the withdrawal effects. Also, we will be collecting the fecal samples 2 days prior to the oxycodone treatment, and after the naloxone injection we will observe the behaviors and we will record them. There will be sniffing, grooming, and rail walking behaviors. Also, the fecal samples will be analyzed using a 16S ribosomal RNA gene amplicon sequencing to look for composition, abundance, and diversity. And also the prefrontal cortex is going to be extracted and analyzed for the presence of pro and mature BDNF. Our behavioral results indicate that the frequency of naloxone-induced behavior is increased in the isolated environment condition, while the enriched environment protects against oxycodone withdrawal. For the microbiome analysis, we can observe a decrease in the relative abundance of Butyridococcus as the oxycodone treatment progresses, and also there are differences seen on the Cholidextribacter and the Rhombustia after oxycodone treatment. Lastly, the BDNF levels in the prefrontal cortex indicate that isolated environment induces a twofold increase in pro and mature BDNF levels in comparison to an enriched condition. So this might tell us that an enriched condition might serve as a neuroprotectant. Thanks for listening. If you have any questions, feel free to email me and I will answer to the best of my knowledge.